A patient steps into a hot shower, and within minutes, their skin erupts in an intense, burning itch. This specific reaction to contact with warm water is a clinical condition known as aquagenic pruritis. Hemopathology links these broad clinical presentations to the physical consequences of specific genetic errors occurring deep within the bone marrow. Accurate diagnosis requires tracing a direct causal line from these chromosomal defects through altered cellular signaling to the macroscopic outcome. We can observe this causal chain by deconstructing three major diagnostic targets. Target 1, polycythemia vera, Target 2, chronic myeloid leukemia, and finally, the B-cell lymphomas. Following these pathways shifts the diagnostic process away from rote pattern recognition, relying instead on a system of mechanical deduction. The primary driver of specific myeloproliferative disorders is the JAK2 V617F mutation. This mutation causes a subcellular failure, locking a tyrosine kinase receptor in an active state. It fires signaling molecules downward. The consequence of this unchecked signaling is panmyelosis, the simultaneous overproduction of red blood cells, white blood cells, and megakaryocytes. This marrow hypercellularity creates specific hazards throughout the body, starting with the white blood cell lineage. As the marrow overproduces basophils and mast cells, these cells accumulate and release massive quantities of histamine. That histamine release is the trigger for aquagenic pruritis. When the patient enters warm water, the temperature change stimulates these excess cells to flood the skin with histamine, causing the severe itching. A parallel mechanical hazard develops from the megakaryocyte lineage, which overproduces platelets. Individual platelets are only 3 to 4 microns across. But in massive, unregulated quantities, they clump together to form microthrombi, physically wedging into and blocking the narrow capillaries of the hands and feet. When these physical blockages choke the capillary networks, they cause the burning pain recognized clinically as erythromelalgia. Itching and burning are not random symptoms. They are the physical and chemical downstream effects of a single broken kinase switch. Large-scale chromosomal abnormalities follow a similarly rigid logic, as seen in the T922 translocation. Sections of chromosome 9 and chromosome 22 physically break and swap places. This reciprocal fusion creates the small Philadelphia chromosome, creating the BCR-ABL fusion gene at the point of connection. Identifying this translocation via a FISH study is the standard diagnostic starting point, but simply confirming the presence of the break is not enough. The BCR-ABL fusion protein's molecular weight determines the phenotype, either 190 or 210 kilodaltons. The 190 kilodalton protein drives acute lymphoblastic leukemia, a severe phenotype with poor prognosis. The 210 kilodalton protein drives chronic myeloid leukemia, presenting a better outcome. A rare 230 kilodalton variant is linked to chronic neutrophilic leukemia, confirming this strict weight-to-phenotype correlation. Cytogenetic diagnosis requires measuring the exact mass of the resulting protein error to effectively determine the patient's long-term clinical trajectory. In B-cell lymphomas, translocations actively hijack existing, highly active healthy promoters to drive malignant growth. The defining molecular driver of Burkitt's lymphoma is the T814 translocation. The mechanical error is precise. The C-MYMOC proliferation gene is cut from chromosome 8 and pasted directly next to the highly active immunoglobulin heavy chain promoter on chromosome 14. Because B cells constantly use the IgH promoter to produce antibodies, this hijacked promoter forces continuous, explosive overexpression of c-MYC, overriding standard cell cycle controls. This genetic overdrive leaves a quantifiable signature on an immunohistochemical K67 stain. Because K67 marks active cellular division, a stain approaching 100% positivity confirms near-total cellular proliferation across the tissue. This relentless proliferation dictates the macroscopic tissue appearance, the classic starry sky histology. The dark sky is the dense sea of dividing tumor cells. The pale stars are tingible body macrophages, desperately attempting to consume the massive amounts of apoptotic cellular debris left behind. We see a mechanical parallel in mantle cell lymphoma, defined by the T1114 translocation. This translocation hijacks the same IgH promoter on chromosome 14, but pulls over cyclin D1 instead, overriding the G1 to S phase checkpoint of the cell cycle. Differentiating these lymphomas requires tracing the pathology back to discover exactly which cell cycle gene hijacked the chromosome 14 promoter. Modern hematopathology reveals a strict, linear logic. We can trace the causal line from subcellular DNA errors through mutated kinases and fusion proteins out to histological stains, and finally to the symptoms the patient experiences at the bedside. Accuracy requires moving beyond the visual patterns seen in histology or physical symptoms alone. Clinicians must identify the exact molecular inflection point, whether that is a specific locked kinase receptor or the precise kilodalton weight of a fusion protein. Every symptom we've explored, from burning erythromelalgia to a lymphoma's starry sky, is a predictable result of a specific identifiable break in a DNA strand.